Next, chromosomes. Chromosomes were first discovered by half-mature scientist. Half-mature scientist in cells of Tradescantia. And the name, the word chromosomes was coined by Valdeir scientist. According to this one, chroma means color, soma means body, that is a colored body. When it was observed, it appears to be like a darkly stained long filament like structure or thread like structure. That is why it was named as colored body or the chromosome. And here the chromosomes are the structures which are called as physical basis of heredity. Because whatever the heredity or inheritance that is occurring in the organisms, that is because physically they are carrying the genes from a parent generation to the offspring generation. That's why these are called as physical base of heredity. And there is one theory is also there, that is called as chromosomal theory, that was proposed by Sutton and Boveri scientists. According to these two scientists, chromosomes are acting as the vehicles of heredity. Chromosomes are acting as vehicles of heredity. So here we know that genes are the structures which are concerned with the inheritance or heredity, and the genes are present on the chromosome. When the chromosomes are moving from the parent generation to the offspring generation, physically they are carrying the genes which are present on them from parent generation to the offspring generation. That's why these are called as a vehicles of heredity according to the chromosomal theory that was proposed by Sutton and Boveri scientists. And as they are physically transferring the genes which are responsible for heredity, that's why we call them as a physical basis of heredity also. So astrocarmine is the one which is helping in staining the chromosomes, so we can differentiate the chromosomes from other cellular contents. Then here the chromosomes are specific for some species, and if you see the size of the chromosomes, generally it is 3 to 12 microns in the length in average in the majority of the living organisms, but in fungi they are of only 0.5 microns. But in Trillium is an plant which belongs to Liliaceae family, which contains the longest chromosomes, that is 30 microns in its length. And when we compare the plant chromosomes with that of animals, plant chromosomes are generally longer than animal chromosomes. Basically, this is the information related to the size of the chromosomes. Then shape of the chromosomes. Generally here if we take a prokaryotic cell like in bacteria, PPLO and cyanobacteria like examples, where it is circular, the chromosomes are circular, naked, and they are made up of double stranded DNA molecule. But in a eukaryotic cell, in different stages of a cell cycle, they are of different shapes. Like in interphase, they are long, elongated, thread-like thin structures, thin and slender structures. But during metaphase, we observe that these are having rod-shaped structures. But during anaphase, when they are moving pulled towards the opposite poles, they acquire different shapes, either V, L, I or J shaped structures will be acquired. Likewise, based on different stages of cell division, they will be acquiring different shapes in the eukaryotic cells, but in the prokaryotic cells they are generally circular, naked and made up of only single double stranded DNA without histone proteins. number. Number of chromosomes will be differing from one species to the other species, but within the species the number will be constant and specific. So likewise number will be ranging from very least number to the highest number. Let us take some examples where number will be deviating. So like name of the organism, the haploid number of chromosomes which are present in them, and the diploid number of chromosomes. Before that, what is this haploid and diploid number of chromosomes? So a vegetative cell or the somatic cell of any living organism contains either single set of chromosomes or two set of chromosomes based on the ploidy of that organism. So here if it contains only, if a cell contains single set of chromosomes, we call it as haploid. So this haploid is represented with small n, or sometimes it may be represented with small x also. Whereas if a cell contains two sets of chromosomes, so we call it as diploid, and it is represented with two digit and small n, or 2 digit and small x. So if these are two general conditions that can be observed in the cells of plants and animals, but rarely we can see triploid condition also, where it is represented with 3n. So where 3 sets of chromosomes are present in a cell. So this type of condition can be seen in the endosperm cells of plants, and also in the alluron layer cells of monocots. Likewise tetraploid, pentaploid, hexaploid, likewise the ploidy is going on. Ploidy means that shows the set of chromosomes inside a cell. So this type of condition will be observed when we consider the number of chromosomes related to an organism or a cell. Organism, haploid number of chromosomes represented as n, and diploid number of chromosomes represented as 2n. Starting with the organism, first one, human beings. In human beings, the haploid set of chromosomes are 23, diploid set of chromosomes are 46. Housefly, that is Musca domestica, the haploid set of chromosomes are 6, diploid set of chromosomes are 12. Rat, haploid set of chromosomes are 21, diploid set of chromosomes are 42. Cat, haploid set of chromosomes are 19, diploid set of chromosomes are 38. Dog, haploid set of chromosomes are 39, diploid set of chromosomes are 78. Fruit fly, that is Drosophila, haploid set of chromosomes are 4, diploid set of chromosomes are 8. Butterfly, haploid set of chromosomes are 190 and diploid set of chromosomes are 380. Likewise, the number is deviating from one species to other species, but the number is the same within the species. That these are some of the examples of various animals where they are showing difference in the number of chromosomes. And in the same way, let us take some examples of plants also where there is a difference in the number of chromosomes from one species to the other species. Let us take the example of Haplopappus. Haplopappus gracilis, which belongs to dicots. Here haploid set of chromosomes are 2, diploid set of chromosomes are 4. Pisum sativum, garden pea, which also belongs to dicot, that belongs to family called as Fabaceae, where haploid set of chromosomes are 7 and diploid set of chromosomes are 14. Next, Allium cepa, that is onion, which belongs to monocot, that is Liliaceae family, where the haploid set of chromosomes are 8, diploid set of chromosomes are 16. Next, Zeamase. Zeamase generally called as maize, which also belongs to Liliaceae family, that is a monocot, where haploid set of chromosomes are 10 and diploid set of chromosomes are 20. Oryza sativa, that is rice or paddy, which also belongs to monocot, that is Poaceae family. Haploid set of chromosomes are 12, whereas diploid set of chromosomes are 24. Nicotiana tabacum, that is tobacco, which is an dicot belonging to Solanaceae family. Haploid set of chromosomes are 24, diploid set of chromosomes are 48. And Gossypium hirsutum. Gossypium hirsutum, commonly it is called as cotton, which belongs to dicot, Malvaceae family. Here haploid set of chromosomes 26 and diploid set of chromosomes are 52. Next, Saccharomyces cerevisiae, that is sugarcane, which belongs to monocot. Haploid set of chromosomes are 40, diploid set of chromosomes are 80. And Ophioglossum. Ophioglossum belongs to pteridophyte, which is a non-flowering plant. Here the haploid set of chromosomes are 630, diploid set of chromosomes are 1260. Likewise, here the number of chromosomes will be differing from one species to other species, but the number will be constant and specific within the same species. Here one more concept had to be known, that is karyotype and ideogram. So karyotype and ideogram talks about the diploid set of chromosomes only. So here the karyotype will depict the diploid set of chromosomes, which explains about shape, or diploid number of chromosomes, which it describes about the size, shape, structure and number of chromosomes present in that particular set. That is called as a karyotype, which gives a description related to the diploid set of chromosomes related to size, shape, structure and number is called as karyotype. And the diagrammatic representation of karyotype is called as ideogram. The same, that is which will give the diagrammatic representation related to the diploid set of chromosomes related to size, shape, structure and number is called as a ideogram. So likewise we can represent the same characters with the help of a diagram called as ideogram. Then coming to structure of chromosomes. So chromosomes, as we said, that they are differing in the shapes and number related to their specific stages and species. So here if you observe the structure, so chromosomes are filamentous structures or in the interphase, filament long thread-like structures in the interphase, whereas in the metaphase where the condensation is maximum, so that is the one which is best site to study the chromosome. The best stage to study the chromosome is the metaphase. Why? Because the thickness will be more and we can clearly observe the structure and structure related aspects related to the chromosome. So here if we start with the structure, so there are many parts related to the chromosome, starting with let us take the basic structure here first. starting the first part, that is the primary constriction. Or we can call this as centromere also. So nearly somewhere in the center of the chromosome we can see a constriction which is formed. This first formed constriction is called as a primary constriction. So primary constriction is the part where the two longitudinal strands of the DNA are attached. So that part is called as a primary constriction. This primary constriction is otherwise called as centromere, and it contains two cup-like structures called as kinetochores on either side, which are proteinaceous in their nature. And because of the kinetochore or proteinaceous structures present in the kinetochore, the spindle fibers will come and implant or attach to the centromere region during cell division. The centromeres will come and attach to the centromere because of presence of kinetochore region in the primary constriction. And during the movement of the chromosomes during anaphase, the chromosomes will acquire different shapes, like as we say V, L, I and J shape. So that is because of bending of the chromosome. So here primary constriction is also useful for bending of chromosome. So these are two characters where we can differentiate the primary constriction from the additional constrictions which are formed. Those are called as secondary constrictions. Then next one is chromatid. We can see the two elongated longitudinal structures. These longitudinal structures are called as chromatids. Elongated, the longitudinal structures present in the chromosome are called as a chromatids, and these two chromatids are attached to each other at the centromere region. And inside these chromatids we can see the presence of DNA along with histone proteins. The DNA is showing the coil-like structure around the histone protein molecules. They form a coiling-like structures which we call them as a nucleosome or chromonema, which form one more structure called as chromonema. So these are nothing but bead-like structures formed inside the chromatids due to the coiling of DNA around the histone molecules is called as chromonema. Nowadays this chromonema is replaced by one more name called as nucleosomes. And one more part is called chromonemata. Chromonemata is the membrane structure which is elongated, or actually it is nothing but chromatid like organization which is present in a DNA structure is called as chromonemata. Secondary constriction. The additional constrictions which are formed in the structure of chromosome other than the primary constriction is called as secondary constriction. So likewise these are called as secondary constrictions. The number of secondary constrictions may be many in a chromosome, and some of the secondary constrictions will give rise to some nucleolar structure here. Thus such type of structure is called as nucleolar organizer. That means some secondary constrictions will give rise to or will help in the formation of nucleolus or nucleoli. Such type of secondary constrictions are called as nucleolar organizers. And secondary constriction contains less stained region. As like the primary constriction, here this is also lightly stained or less stained region. And how can we differentiate the secondary constriction from primary constriction? Means primary constriction contains kinetochores, but the secondary constrictions will lack the kinetochores, and primary constriction will help in bending of the chromosome, while the secondary constriction does not help in the bending of the chromosomes. So likewise we can, and one more thing, the nucleolar organizer is present in the secondary constriction which will help in the formation of nucleolus, whereas that nucleolar organizer is absent in the primary constriction. Based on these characters, we can differentiate the primary constriction from the secondary constrictions. Then satellite or tribent. Sometimes we can see some spherical shaped structure beyond the secondary constriction. Some spherical structure beyond secondary constriction is called as satellite. It is otherwise called as tribent also. So it is not compulsory that all the chromosomes should contain satellite, but the chromosomes which contain satellite are generally called as SAT chromosomes. SAT means sign acid thymonucleinico. So that means the part of the chromosome which generally lack or contain less amount of thymonucleic acid. This type of structures are called as satellites, which are present in some of the chromosomes. Then next one is telomere. Telomere is the tip of the chromosome. The extreme tip of the chromosome is called as telomere, like this part or this part. These are called as a telomeres. Then here telomeres, because of telomeres, polarity is the chromosomes will exhibit polarity due to presence of this telomere, and that never the two tips of two chromosomes will unite. Because of the polarity, never the two tips of the chromosomes will unite. But when a broken pieces are present, the broken pieces of the chromosomes will unite with each other, because that part does not contains the telomere. Likewise, these are the some basic information are related to the structure of the chromosome. The classification of chromosomes. Chromosomes are classified based on various criteria. The first one is based on the functions. Based on functions, the chromosomes are classified into autosomes and allosomes. Autosomes are the chromosomes which will determine vegetative characters of an organism, whereas these are not related to sexual characters, or these are not concerned with the determination of the sexual characters of an organism. And allosomes, so these are the one which will determine sexual characters of an organism. When we compare the number of autosomes with that of a number of allosomes, generally the number is more in the autosomes. Number of autosomes is more. Generally here the number of allosomes will be comparatively less than that of the number of autosomes. Let's take the example of human beings, where the diploid set of chromosomes is 46. That is 2n is equals to 46. Out of that, some are autosomes and some of them are allosomes. Autosomes are 44 in their number when it is considered as diploid set of chromosomes. When it is haploid set, it is 22 in their numbers. And allosomes, if it is diploid set, they are only 2 in number. So 24 plus 2, it comes to total of 46 chromosomes. And if it is a haploid set, it is only 1. That is 22 plus 1, that is 23. So 2n is equals to 46, whereas n is equals to 23. So this is a total number of chromosomes present in the human beings. Likewise, there is a difference between the autosomes and allosomes based on the function, which character they are determining for that particular organism. Next criteria, that is classification of chromosomes based on number of centromeres in a chromosome. First one, monocentric. Monocentric means the chromosome which is having only single centromere. So this is the most common type of chromosome that is present in the living organisms. Second one is dicentric, where one chromosome is with two centromeres. One chromosome is with two centromeres, that is called as dicentric chromosome. Third one is polycentric chromosomes. Polycentric means if many centromeres are there, if many centromeres are present in a chromosome, we call it as a polycentric chromosome. And there are some chromosomes where there is no centromere at all. So here centromere is absent. Actually, we know that spindle fibers will come and implant or attach to the centromere part due to presence of kinetochores. But in eccentric chromosomes, the centromere is completely absent, means the kinetochores are also absent, so that this eccentric chromosome does not participate in cell division. Why? Because the spindle fibers can't attach to the eccentric chromosome due to absence of primary constriction and the kinetochores. That is the reason why eccentric chromosome can't be participated in a cell division process. So these are four types, are based on the number of centromeres present in a chromosome. Monocentric, as we say, it's the most common type. So that is further divided into four types based on the position of the centromere in a chromosome. Based on position of centromere, monocentric chromosomes are further classified into following types. First one is metacentric. Metacentric means the chromosomes in which the centromere is present in the center, or centromere is present in the middle of the chromosome. So here if it appears to be like this, where this is centromere, these are the arms which are present in the chromosome. So this total structure is called as chromosome. These are the arms. Here in this diagram, we can see the centromere is present exactly in the center, such that the two arms are equal in their length. And when the chromosomes are pulled in towards the opposite poles during anaphase, so the chromosome will acquire V shape. And two equal arms are present here. The second one is submetacentric. Submetacentric is the one where centromere is present near to the centromere. Centromere is near to the center, but it is not exactly in the middle as like in the metacentric. So due to this, we can see that the arm size will be little bit differing. So it is a small arm and this one is a large arm. One will be small and one arm will be small and one arm will be large. So this is a centromere. during anaphase, when the chromosomes are pulled toward opposite side, the chromosome will acquire nearly L-shaped structure with one long arm and one short arm. So it appears to be L-shape. The third type is acrocentric. Acrocentric is the type of chromosomes in which the centromere is present near to the tip. Is not exactly at the tip but near to the tip of the chromosome. So because of this one of the arm size will become very long and one of the arm will become very short. So very short arm and very long arm. So here it is a centromere. Because of this one, when the chromosomes are moving towards opposite poles during anaphase, it will acquire J shape, the very long arm, centromere with a short arm. So we can show like this, it is a J shaped structure. Then the fourth one is telocentric. This is a type of chromosome where centromere is at tip of the chromosome. This is a condition where the chromosome is like this. This at the centromere part, at the telomere part, the centromere is present. At the telomere part, the centromere is present. So this is an arm. So here in this diagram we can see that telocentric chromosome contains only single arm because the other side of the centromere does not have any arm there. So that's the reason why it appears to be like I-shaped structure. So which is having single arm. So likewise, based on the position of centromere, the monocentric chromosomes are classified into 4 types.